encompassing was using computational analysis to observe the genome of microbe Zymomonas mobilis. So in this project, it was a bit different than traditional projects in the sense that there was no new data that we made, right? Like there was no new experiments that we did to make new data. Instead, what we did was we had we used data from two separate studies, right? And we analyzed those data sets, right? Those large data sets, and we parsed through them and we analyzed them basically to see if there's any patterns that appear, right, from these data sets, or if the data is agreeing with each other, right? If they're not agreeing with each other, and kind of analyzing the context behind the data and kind of reading in between the lines, right? So the two studies, the two data sets we've taken are from two studies, right? The first of which is called the EMBO study, in which it was mainly focused on Zymomonas mobilis, or Z mobilis, as a biofuel, right? And it did gene annotations for Z mobilis in plenty of experimental conditions, mainly based on plant hydrolysis, right? To test its ethanol efficiencies, right? And it's to see what genes are triggered due to these different ethanol stresses, right? The second study was the JBAC study, right? And the JBAC study was a more comprehensive study based, it's a more comprehensive study based on actually annotating the entire Z mobilis genome, right? Using a new technique called bar-coded transposon mutagenesis, right? And it was a lot more comprehensive than the first study because it was mainly focusing on actually annotating those genes, right? So, moving on. So, why the big thing about these two studies, or the big unifier, is the microbe itself. Z mobilis. Why Z mobilis of all things? Well, Z mobilis is actually used industrially has plenty of industrial uses, and is used industrially simply because it is known to have a high tolerance to ethanol and can produce ethanol, right? In addition to that, specifically for the JBAC paper, Z mobilis has the smallest genome of all the bar-coded transposon mutagenesis technique bacteria, right? So all the bacteria that had, bar-C, or bar-coded transposon mutagenesis, done to it, Z mobilis was the smallest, right? And thus, doing such a comprehensive and large-scale approach to identifying and annotating its genes would be more feasible, right? And would be a little bit easier than doing it with E. coli or something with a larger genome, right? So moving on to motivation and approaches, right? Moving on to motivation and approaches. So both the studies are really do really well together, but the main thing is that the EMBO study was kind of like a scaffolding for the JBAC paper, right? The JBAC paper does a really thorough job annotating the gene functions, right? And it identifies the significant phenotypes and compares the fitness of those genes to the weaker genes with weaker phenotypes in order to further annotate it, right, with confidence. But in order for it to do that, they actually used data from the first EMBO paper, right? Even though that paper was mainly focused on Z mobilis as a biofuel, and used different ethanol stressors for it, right? And only about 58 experiments, right? What's it called? The JBAC paper kind of built upon that. And instead of 58, it was actually, the JBAC paper used 492, right? Using kind of the same method and the same, gene fitness method. And so looking closer on the table itself, right, you see a lot of blues and yellows and a bit of black, right? So those blue parts basically mean that the gene that they found has reduced fitness under that stressor. And so what is fitness, right? What does it mean to have reduced fitness or positive fitness, right? And so reduced fitness is basically, or fitness itself, as characterized by the EMBO, both the EMBO and the JBAC paper, is basically just log2 change in the abundance of strains with insertions in the gene, right? So the negative values, right? So those blue indicators, the negative values indicate that the gene is beneficial for fitness, and that strains with the mutated gene, right? Those insertions that knock out those genes have reduced fitness, right? So if that gene is mutated, that gene is knocked out, or something happens to that gene, in those stressors, the growth of Z mobilis is inhibited, right? And so if you have enhanced fitness after a knockout or after a mutation in that gene, thus you know that that gene is not necessary for growth and actually inhibits growth in that stressor, right? So it's kind of back and forth. So I find it super interesting that they use the same approach of the JBAC paper used the same approach as the EMBO paper, right? So moving on to the main learning section. So on the right, there's a heat map of basically the 1586, so 1,586, these mobilist genes done across 492 experiments, right? And these experiments were way broader than the EMBO paper, right? They included more than just ethanol stresses. Right? They put a bunch of different types of antibiotics as a stressor. They put different medias as a stressor, just kind of seeing that broad range of stressors to see what genes activate and what genes don't, right? What genes show up, what genes don't, right? And so theirs is a lot more comprehensive, and basically, again, that the blue and the yellow and the black is still the same. If you see a large amount of blue, that means if that gene is knocked out or if that gene is mutated, then that gene is vital for Z mobilis's growth in that certain experiment or that certain stressor, right? So in this, we have a lot more black, right, and a lot more experiments. So it's a lot more black. So what black means is just there's a lot of weak phenotype but significant genes, right? They have weak fitness, but they still have a phenotype very significant, right? So in order to kind of further analyze those weaker genes and detect them, the JBAC paper did long-term growth experiments, right? So now if we look at figure 2, what I really want everyone to key in here is this x on the x-axis, the -1, 0, and 1, right, on the x-axis, right? So that means after 3 days, these are kind of weak, right? They're not really very prevalent, right? Not greater than 1, they're not greater than the absolute value of 1, right? They're actually less, so they're kind of weak, right? They're not really, it helps a bit or it doesn't help a bit, right? And so what we want to see is, okay, looking up from the on the y-axis, so after 7 days, right, after longer time, these growth experiments happen, right, over 7 days instead of 3 days. Do these genes change? And yes, a lot of them do, right? So we look up here in this little area, a lot of these genes now become greater, right? So we're looking here. So a lot of these genes actually become greater than -1, right? They're becoming more significant, right, and easier to detect over time, right? Meaning that these weak phenotypes are significant, right? We just needed more time. They just needed more time to show or to express themselves, right? So in the end, why is this important, right? Why even talk about this, right? So the JBAC study itself is super important because it presents the framework of the future of functional annotation of bacteria utilizing bar-coded transposon mutagenesis to get data-driven high-throughput data, right? So it's very thorough, right? And it's super important because it prevents a possible solution to the challenge of us figuring out the functions of sequenced genes, of millions of sequenced genes that we don't really know about, right? And though there is some points of, debate or talk, such as, using Z mobilis, a bacteria with small genome, well, with a small genome, how can it be further like, is it as comprehensive as it seems because it has a smaller genome? So how about a bigger bacteria? Is it as thorough as it is? You know, and it is, right? And it is, and it has, this technique has been applied to other bacteria, right? So this kind of, these papers kind of built the framework on future gene annotations. So yeah. Um, and so for the acknowledgements, I'd like to thank Professor Arash Jamshidi and the CalTeach program for allowing me to take part in this opportunity in this project. And I would also like to thank Professor Ashish Mishra for being my research mentor during this project, and Andrea Mukhopadhyay for offering additional guidance on this project. Thank you.